Welcome to my channel, Pure Biology, and this is today I come with another video, that is the point mutation. Point mutation is the changes of base pair in the DNA or gene, and due to the changing of the base pair in the DNA, some mutations is possible, or the variations or the some variable characters or the features that is possible. So before that I explained about the chromosomal mutation, and you just check out it in previous video what is the chromosomal mutation and what changes occur in this chromosomal mutation, and after this you just watch this one, the point mutation. Okay, so what is the point mutation? Point mutation is the sequence of base pair is altered. Okay, sequence of base pairs is altered. Here single base pair is changing in changing or altered, or sometimes a sequence of base pair that changes. Clear? So here, and the mutation is possible, sometimes addition of some base pair or the deletion of some base pair. Clear? So here this is the mutation or point mutation, where we say that the base pair is altered. Okay, one base pair or the sequence of base pairs altered. And here two kinds of mutation is seen. One is the somatic mutation, another is the germline mutation. The somatic mutation is when the mutations occur in the somatic cell and the changing is found in the somatic cell, that is the somatic mutation. And germline means the gamete cell, okay, or the germ cell. Some base pair that changes in the germ cell, and that's why some features added or deleted in the from the gamete cells. Clear? Now, two kinds of mutation is possible. First, we understand about this. This is a substitution and another is the frame shift. What is the substitution? Substitution means here the changes of one base pair to other. Okay, changes of one base pair to other. And this type of substitution process generate two kinds. We see here. One is the transition and another is the transition and the transition and transversion. I think so. One second. Transition and transversion. Okay, transition and transversion. This is the transversion. Transversion. Okay. This is transversion. Okay. The first one is the transition, where the base pair, one pyrimidine base that changes to the other pyrimidine base, or the one purine base that changes to other purine base. Here the changing is possible within the same base pair. You know all about what are the purine bases and what are the pyrimidine bases, AT and GC. AT is the purine base and GC pyrimidine base. So here the changes is possible within the pyrimidine to pyrimidine or the purine to purine. But in case of the transversion, here the purine to pyrimidine, pyrimidine, or pyrimidine to purine, pyrimidine to purine. Clear? This is the transversion. So this is substitution process. Substitution process, we find out that the two kinds of mutation is possible. One is the transition, another is the transversion. Transition means pyrimidine to pyrimidine or the purine to purine, but in case of transversion this is altered. That means purine to pyrimidine or pyrimidine to purine. Here. Now second one we read that the frame shift mutation. Okay, frame shift mutation means addition or the deletion of the base pairs. Okay, addition or the deletion. Here I just give one example of addition but not the deletion. You just understand, here this is the example of addition, like that the deletion process is same way the deletion process is occurred. Okay, so here this is you think this is first of all I just rub it. Okay, I just rub it. Okay, you see this is the sequence of base pairs. Okay, sequence of base pairs, and from this one mRNA that continuing this is formed. Okay, I prime this mRNA is formed. And what happened if the addition of one base pair is possible or is occur. You just see here T, the normal mRNA chain is like that, but after mutation, this is possible. That is here A, and I just mark this position, this position, okay. Here one entry of T is seen, okay. One entry or one addition of T base pair is seen between the C and G, and then what happens there? After addition of this T base pair, here another base pair A is formed, that is its complementary base pair, and when the mRNA is formed, its complementary base pair is U. You know that about DNA ATGC, and in case of RNA ATUC, uracil, okay. So here complementary base pair of A is uracil. You see here uracil is absent here, okay. Uracil is not seen because the addition of T is not occurred in that situation. But after when the mutation is occurring, they are conjugating, then addition is possible, and the complementary base pair of T is A, and the complementary base pair of A is U in case of mRNA. So the total changing of the mRNA is seen in such place, okay, in such place. This is the frame shift, okay, frame shift. So first one is the substitution, second one is the frame shift. Next we learn about the missense and the nonsense. Missense means here the base pair change causes a change in an essential amino acid. You all know that in translation, after formation of the mRNA, in process of transcription, the translation is occurring, and there the protein synthesis or the essential amino acids are formed or produced that are very important to our body. This essential, because if one essential protein, what is that? Amino acid. Essential amino acid is changing in anyhow or any cases, then some disorder is seen in some cases. Clear? So every amino acids are very important for our body. If one change, any change is possible in the amino acid groups, so for the mutation, and that's why disorder may be seen in the body. Clear? So this is the example of missense mutation. Okay, this is missense mutation example disorder, that is sickle cell. Sickle cell, you know sickle cell here, the shape of the generally hemoglobin and the red blood cell, the shape of the red blood cell is circular like that, but in case of sickle cell, the sickle shaped. Clear? Just changing of one amino acid, essential amino acid, it is created. Clear? So what happened is the glutamic acid is very important for the forming of the RBC, obviously in sphere shape. But in place of glutamic acid, when the valine is formed, okay, when the valine is formed, the structure of the red blood cell changes to the sickle shape. Clear? This is called the sickle cell anemia. Okay. So here GAG, its complementary bases are CTC, and when mRNA formed, that is GAG, and it is the actual mRNA when the essential amino acid is glutamic acid. But when the mutation is occurring, that is GAG, here addition of something or any changes, A is altered with T, then its complementary changes A here, and its mRNA also changes. Its mRNA is GUG, okay, GUG, this is GUG, okay. And what happens that the valine is the essential amino acid that is formed. But valine is not important here or not needed here for the production of the or for the structure of the red blood cell. Here it is here needed the glutamic acid, but for the mutations, and it is a missense known as the missense mutation, just one base pair here changes, and glutamic acid that changes to the valine. Next one we learn about the nonsense. What happened in the nonsense? There causes the premature termination of the polypeptide chain synthesis. If the nonsense mutation is occurring, then what happened? Actually, the total the polypeptide chain, normal polypeptide chain, that is terminated and that is shortened. Okay, that is shortened. Okay, so what happens in the nonsense codon? We know all the termination codon. 3 termination codons are there. When the termination codon attach to the protein during the protein synthesis, then polypeptide chain that is continuing that becomes stop. So the formation of the polypeptide chain stop just when the any one of the 3 termination codon that attach in the place of the chain, in any position of the polypeptide chain. Okay, so here in case of nonsense, new termination codon is arised. Okay, new termination codon is arised. Generally mRNA actually knows proper 3 termination codon. Okay, if there is any changes occur in the termination codon or the mutation of the termination codon is occurring, then what happens? The mRNA cannot identify the particular termination codon, and at that time when the mRNA cannot identify the codon, then it stops its forming of the polypeptide chain. Clear? So here what happened here? The polypeptide chain become shortened. Okay, the actual length of the polypeptide chain, that is very important for the formation of the essential amino acids. But when the polypeptide chain becomes shortened, then what happens? That becomes non-functional. Okay, that becomes non-functional. So these are all the about the mutations. We learn after that again another type of point mutation. Okay. Okay, now we learn about the silent and the neutral mutation. What is the silent mutation? The changes of base pair in the gene but altered codon in the mRNA specifies the same amino acid in the protein. Okay, any changes in the codon but that is not effective. That's why it is silent. That is not effective for the production of the same type of amino acid. Okay, here the transition mutation is occurring. You see that AT transfer to GC transition mutation is occurring here, and in case of transition mutation, when AT transfer to GC, the codon transform AAA to AAG. Clear? But both the codon that specifies the lysine amino acid. So you cannot recognize there is already a mutation is completely that means possible not the mutation is okay. Once again, sorry for the disturbance. I just stop this. So, in case of silent mutation, here the transition of the base pair is we see here the transition base pair mutation is possible here, and AD transfer to GC, and here the codon is formed AA to AAG. Okay, but both the codon AA and AAG that specifies lysine. Okay, that specifies lysine. And in case of neutral, you see here that the changes of the codon and the resulting amino acid substitution produces no detectable change in the function of the protein translation. Here also transition point mutation is seen, and AAA transfer to AGA, but here this codon recognize lysine and this codon recognize the arginine. Clear? But here we also cannot recognize the mutation is already happened because both have the similar properties. Clear? Here this is silent, this is the neutral. In case of silent, both the codon, they specifies or they recognize the same essential amino acid. So silent mutation we cannot identify. In same case, neutral mutation also, this point mutation we cannot identify because already mutation happened here, but this lysine, this recognizes the lysine and this recognizes the arginine, but both have as usually the similar properties. Okay, both have the similar properties, so the protein chain has not been affected. Okay. So this is the silent and the neutral. In both the cases, the protein chains not be disturbed. Okay, or the characters of the organisms also in some cases not be changeable or it is not seen. And in case of reverse mutation, here this is two type of changes is possible. One is the forward changes and another is the backward changes. Clear? And two types of reversion we have seen. One is the true reversion and another is the equivalent reversion. True reversion means back to the wild type amino acid. Means, example, we just follow the example. In case of AAA, this forward mutation is occurring here and this is GAA. And you see here, when AAA codon, this recognizes lysine, and when GAA, this recognizes the glutamic acid. But when the this again mutation, here the mutation again occurring, then GAA changes to AAA. Complete AAA codon is similar to the wild type codon. You see here, AAA codon, that is previously we have seen or previously we found that AA codon is there, and when the mutation that changes AAA into GAA, but when again mutation is occurring, that changes to the previous wild type codon AAA. The codon or nucleotide, changing of the nucleotide is not seen here. Okay, nucleotide is same. So first one is recognized that recognizes lysine. This recognizes glutamic acid, and again AAA that recognizes the lysine. So nucleotide as usually same. That's why this is the true reversion. Okay, this is true reversion. But you see the second example when UCC. One second. UCC, that is SER. UCC, that is SER. UCC, that is that changes to after mutation UGC. When SER was the amino acid, when it after mutation changes to UGC, the cysteine, it is the one amino acid that recognized by the UGC. And again, when the mutation is seen, it changes to AGC. Then again ACR, serine. Okay. So here you see that first one nucleotides are UCC, but after second mutation, you see that AGC is the nucleotides. Okay, codon. So codon is not same. You see here codon. In first case the codon is same, but here you see the codon is not same. But both the codon they recognize the ACR or the serine. Okay. So this is the equivalent reversion. Why equivalent reversion? Because different nucleotides. Okay, different nucleotides. Here the same nucleotides, but here you see the different nucleotides. So the function also maybe changes or not changes, but depending on the mutation. Okay, that means the partial reversion is seen here. You see that is it is a complete reversion, but here the partial reversion is seen here, and the feature may be changes or not changes, but depending on the character or the not character, not is the base pairs obviously, or the codon. Clear? So this is the reverse mutation, and one mutation we last one that learned here, that is the suppression mutation. Okay, now we learn about the suppression mutation. Here what happened, the effects of the mutation may be diminished which occurs at different site from that of original mutation. That means it explains, suppose mutation of one normal gene is occurring, okay, and after mutation the mutant feature that is also arising. Clear? But due to the suppression mutation, what happened? The character of the mutant gene that will be suppressed. Okay, that will be completely suppressed. There is one example, or it occurs in generally same gene but different site or different genes. Okay. So suppose this is the example. Here the wild type gene, and that forms the wild type protein. Suppose wild type protein, it has to be formed, okay, wild type protein, but due to mutation some mutant protein is formed, okay, some mutant protein is formed. But when the suppression mutation is continuing or this occurring in over the mutant protein or over the formation of mutant protein or over the gene, mutant gene, what happens? The wild type protein again recognized. Okay, the wild type protein is recognized by the codon. Suppose this is example 3' AUG 5', that is for tyrosine recognition. Okay, its complementary base pair must be that UAG 5', 3'. Okay, and here the mutation is occurring, and after mutation is occurring, then the codon that will form in this here in this during the protein synthesis, but this codon obviously recognize tyrosine again. Okay, tyrosine again. So the mutation is AUG. This mutation here, the mutation is occurring here, and as a result the 3' AUC is generally formed, but nonsense code, it reads the nonsense code AUAG. It reads the nonsense code, okay, and that is generally recognized the tyrosine. So the feature is not changing here. Feature is not changing here due to the mutation, because the totally new feature or the mutant feature that is suppressed by the suppression mutation. Actually, the essential amino acid, that is the tyrosine, that must be recognized with its complementary basis. But here, after the mutation and suppression mutation, the codon that we form, that can also recognize the tyrosine. So here the protein chain formation not be disturbed. Clear? And one thing is that intragenic and intergenic I explained here also, but now I also explained that suppression mutation is generally possible in the same gene or the different gene. If it is occurring in the same gene but the different site, that is it is called the intragenic. And when this suppression mutation is occurring in the different gene, then it is called the intergenic. Clear? And now we learn about the spontaneous and induced mutation. Spontaneous mutation means the naturally occurring mutation. This is not the accidentally occurring mutation, but induced mutation generally this occurred by the accidents or by some agents. Okay, like the mutagen. Sorry. And spontaneous mutations, one type, this is called the deamination, and under deamination this is depurination and the depyrimidation. Okay, deamination means the removal of the amino acids. Sorry, once again, removal of amino acids. Sorry, this is not removal of amino acid, this is the removal of amino group. I have a doubt, and that's why I check and then explain. Removal of amino group. Clear? So here you see that cytosine that changes to uracil. Clear? And there are two kinds. One is the depurination and another is the depyrimidation. In case of depurination, here the loss of purine due to the breakage of the glycosidic bond, and in case of depyrimidation, here the loss of pyrimidine base. Clear? This is the spontaneous and the induced mutation. Another two mutations are there. One is the transposition mutation and the oxidative damage. That is due to the attack by the reactive oxygen, the species must be considered as the major source of the spontaneous damage. Like that same, that is you say that it is one kind of induced, because here the actually the role is played by the reactive oxygen. Okay, so what happened here, the attack by the reactive oxygen to the species, the mutation is created, and this is called the mutation that is occurring by the oxidative damage. And the last one is the transposition. The transposition mutation means we know about the transposable elements. Okay, the transposable elements, they have a feature, they can easily insert inserted any place of the gene. Okay, and it can move along the gene. Okay, when I explain about the transposable element, then I tell about that. Today I just explain briefly that transposable mutation. So what happened here? The transposable element when inserted in a place that is not necessary for entering the transposable elements, but it has a capacity that it can insert in any places. So when it insert a place of the gene that is the place is not needed for the entry of the transposable element, then the mutation is created. Okay, so this mutation is called the transposition mutation. Okay, so today point mutation I just completed. Okay, and in my next video I will explain like that another topic.